This video tutorial is all about muscular contraction. Specifically, we're going to look at the sliding filament hypothesis. Myofibrils are contractile units of skeletal muscle and contain two types of protein filament. Thin filaments, which are aligned to make up light band, and these are held together by the Z line, and thick filaments, which make up the dark band. The thin and thick filaments overlap, but in the middle of the dark band, there is no overlap. This is called the H zone. The distance between two Z lines is called a sarcomere. This is the functional unit of the muscle. At rest, a sarcomere is about 2.5 micrometers long. The thick and thin filaments are surrounded by sarcoplasmic reticulum. The thin filaments are actin. Each filament consists of two chains of actin twisted around each other. Wound around the actin is a molecule called trypomyosin, to which are attached globular proteins of troponin. Each troponin complex consists of three polypeptides. One binds to actin, one to trypomyosin, and the third binds to calcium when it is available. Trypomyosin and troponin are part of the mechanism to control muscular contraction. At rest, these molecules cover binding sites to which the thick filaments can bind. Each thick filament consists of a bundle of myosin molecules. Each myosin molecule has two protruding heads, which stick out at each end of the molecule. These heads are mobile and can bind to the actin when the binding sites are exposed. During contraction, the light band and the H zone get shorter. Therefore, the Z lines move closer together and the sarcomere gets shorter. This observation led to the sliding filament hypothesis. During contraction, the thick and thin filaments slide past one another. The sliding action is caused by the movement of the myosin heads. When the muscle is stimulated, the trypomyosin is moved aside, exposing the binding sites on the actin. The myosin heads attach to the actin and move, causing the actin to slide past the myosin. We're now going to look at the control of contraction. So when the muscle is stimulated, the action potential passes along the sarcolemma and down the transverse tubules, or the T-tubules, into the muscle fiber. The action potential is carried to the sarcoplasmic reticulum, which stores calcium ions and causes the release of calcium ions into the sarcoplasm. The calcium ions bind to troponin, which alters the shape, pulling the trypomyosin aside. This exposes the binding sites on the actin. Myosin heads bind to the actin, forming cross bridges between the filaments. The myosin heads move, pulling the actin filament past the myosin filament. The myosin heads detach from the actin and can bind again further up the actin filament. Millions of cross bridges can be formed when the actin and the myosin filaments between them. Once contraction has occurred, the calcium ions are rapidly pumped back into the sarcoplasmic reticulum, allowing the muscle to relax. ATP supplies the energy for contraction. Part of the myosin head acts as an ATPase and can hydrolyze the ATP to ADP and an inorganic phosphate, releasing energy. The myosin head attaches to the actin filament, forming a cross bridge. The myosin head moves or tilts backwards, causing the thin filament to slide past the myosin filament. This is known as the power stroke. During the power stroke, ADP and the inorganic phosphate are released from the myosin head. After the power stroke, a new ATP molecule attaches to the myosin head, breaking the cross bridge. The myosin head then returns to its original position, so it swings forward again, as the ATP is hydrolyzed, releasing the energy to make this movement occur. The myosin head can now make a new cross bridge further along the actin filament. As there are millions of myosin heads involved in muscular contraction, there is a huge requirement for ATP. The ATP available in muscle tissue is only enough to support at most 1 to 2 seconds worth of contraction. ATP, therefore, must be regenerated very quickly in order to allow continued contraction. There are 3 mechanisms involved in maintaining the supply of ATP. Firstly, aerobic respiration in mitochondria. Muscle tissues contain a large number of mitochondria in which aerobic respiration can occur, and the Bohr effect helps to release more oxygen from the hemoglobin in the blood. However, during intense activity, the rate at which ATP can be produced will be limited by the delivery of oxygen to the muscle. Secondly, anaerobic respiration in the sarcoplasm of the muscle tissue can occur. Anaerobic respiration can release a little more ATP from the respiring substrates. However, it leads to the production of lactate or lactic acid, which is toxic. Anaerobic respiration can only last a few seconds before lactic acid buildup starts to cause fatigue. And finally, creatine phosphate. Creatine phosphate in the sarcoplasm acts as a reserve store of phosphate groups. The phosphate can be transferred from the creatine phosphate to ADP molecules, creating ATP molecules very rapidly. The enzyme creatine phosphotransferase is involved. The supply of creatine phosphate is sufficient to support muscular contraction for a further 2 to 4 seconds.